Okay, imagine this. You have to copy a library, not just one book, but thousands of them, word for word, with almost zero mistakes. And you have to do it all in like a few minutes. Sounds impossible, right? Well, that's exactly the challenge your cells face every single time they divide. So how on earth does life pull off this amazing feat? Let's dive in.
So, to really get the scale of this, you have to remember our DNA has billions of these little chemical letters. Now, let's say the copying machine inside our cells was just 1 in a million accurate. I mean, that sounds pretty good, right? Well, with that error rate, you'd still get 6,400 mistakes every single time one of your cells divides. I mean, that's a total recipe for disaster.
But here's the crazy part. The reality is, it's not 1 in a million. The actual error rate is less than 1 in a billion. Our cells are just unbelievably precise. To put that in perspective, it's like typing out an entire encyclopedia set 100 times over and only making a single typo. It's just mind-boggling.
So, that's the big question, isn't it? How is this even possible? How does this little molecular machine achieve that kind of breathtaking speed and just staggering precision? Well, let's unpack the secrets of how DNA gets copied.
You know, before we could even get to the machinery, scientists had to solve the first big puzzle. What's the basic strategy here? How does the process even work? It turned out to be a real scientific detective story.
All right, so there were 3 main ideas on the table. The first, semi-conservative, suggested that the original DNA unzips and each old half acts as a template for a new half. Then there was conservative, which imagined the original DNA staying completely intact while a totally brand new copy gets made. And finally, dispersive, which was the idea that the original gets kind of chopped up and sprinkled throughout both of the new copies. So, which one was it?
Well, this beautiful, elegant experiment in 1958 totally settled the debate. What they did was they grew bacteria with a heavy version of nitrogen, which made all its DNA really dense. Then they switched it to a medium with light nitrogen. So after 1 generation, they checked the weight of the DNA, and all of it was perfectly in the middle, an intermediate weight. Bam! Right there, that completely ruled out the conservative model. But the real clincher came after the second division, when they found 2 separate bands, one intermediate and one light.
And that result? It was a perfect match for what the semi-conservative model predicted. Each new DNA molecule was a hybrid, one old heavy strand and one new light one. And just like that, the first big mystery was solved. DNA replication is semi-conservative. Case closed.
Okay, so we know the basic strategy now, but who or what is actually doing all the heavy lifting? Well, it's time to meet the all-star team of enzymes, the tiny molecular construction crew working inside your cells.
First up, we've got helicase. The easiest way to think about this one is as the unzipper. Its whole job is to just latch onto the DNA and race down the line, breaking all those little bonds between the bases and prying the 2 strands apart. This creates something we call a replication fork.
Next on the team is primase. Now, here's a funny thing. The main builder of this crew has a weird quirk. It can't just start building on a bare strand of DNA. It needs like a starting block. That's where primase comes in. It's the enzyme that builds that little starting block, a short strip of RNA that we call a primer. And now for the star of the show, DNA polymerase. This is the master builder. It comes in, latches onto that RNA primer, and just starts flying down the template, adding new DNA bases and building the brand new strand. But get this. It's not just a builder, it's also its own quality inspector. It actually proofreads its own work as it goes, catching and fixing mistakes on the fly. Incredible!
But of course, there's a complication. DNA strands are what we call anti-parallel, meaning they run in opposite directions. And it turns out DNA polymerase can only build in one direction. So this means one strand, the leading strand, gets built nice and smooth all in one continuous piece. But the other one, the lagging strand, has to be built backwards away from the unzipping action in these short, disconnected chunks called Okazaki fragments.
And this is where our final crew member comes into play. DNA ligase. You can just think of this one as the gluer. Its job is to come in behind everyone else and seal up all the little gaps between those Okazaki fragments, stitching them all together to create one final continuous strand of DNA.
Now, it's worth pointing out that this whole process isn't exactly the same for, say, a simple bacteria versus a complex organism like you or me. Bacteria have this single starting point on their circular chromosome, We eukaryotes, on the other hand, have tons of starting points on our long, linear chromosomes. And that one difference, circular versus linear, creates a really unique and fascinating problem.
So this brings us to a new, kind of suspenseful twist in the story. It's a problem that only happens in organisms with linear, string-like chromosomes, like us. It's called the end replication problem. Just think about it for a second. On that lagging strand, that very last RNA primer, the one at the absolute tip of the chromosome, eventually gets removed. But because of that directionality issue we talked about, there's no place for DNA polymerase to land and fill in that final little gap. So what happens to that tiny empty space at the very end of our DNA?
The stark answer is nothing. That gap just can't be filled. And the result? Every single time one of our cells divides, our chromosomes get just a tiny, tiny bit shorter. And over a lifetime, well, that starts to add up.
I mean, losing a piece of your genetic code every time a cell divides sounds like a pretty catastrophic design flaw. But life, as it always does, has come up with an ingenious, though maybe not totally perfect, solution to this ticking clock.
Say hello to telomeres. The best analogy for these are the little plastic tips on the end of your shoelaces. They're called aglets. Telomeres are just like that. They're these long, repetitive strings of DNA, TTAGG, over and over and over, at the very ends of our chromosomes. They don't actually code for anything important. Their only job is to be a disposable buffer that protects the real genes from getting lost during that end-shortening process.
So how do we maintain these protective caps? Well, with a special enzyme called telomerase. It's a really unique kind of polymerase that can actually add those repetitive telomere sequences back onto the ends of the chromosomes, basically rebuilding the buffer and fighting back against the shortening. But there's a catch, and it's a really big one. While our very important stem cells have active telomerase, most of our regular body cells, our somatic cells, turn the enzyme way down or even switch it off completely. And what that means is, for most of the cells in our body, that clock is still ticking down. The telomeres are getting shorter and shorter with every single division.
And this is where it all connects right back to our own health. This slow, steady shortening of our telomeres is a major driver of cellular aging. When they get too short, cells just stop dividing. But here's the dark side to this whole story. Some cells can figure out how to flip that telomerase switch back on. This lets them become effectively immortal, completely bypassing the normal aging clock and dividing endlessly. And we have a word for that: cancer. In fact, it's a strategy that about 85% of all human cancers use.
So we end on this final thought. It seems our bodies have this built-in clock that causes us to age, and it's probably there as a defense against cancer. But the main survival strategy for cancer is to figure out how to break that very clock. It really reveals this deep, profound biological trade-off right at the heart of our existence, this delicate balancing act between growing old and the ever-present risk of uncontrolled growth.